Good morning here, at least. I think I'm going to speak for a very short time because I have other meetings going on today, and I sent videos that Eric can play whenever he wants. My name is Barend Mons. I'm one of the judges, and I'm originally a biologist by training, molecular biologist, but working for many years already on machine-assisted hypothesis generation. Also, the one of the co-founders of the FAIR principles that is now also rebaptized from the original meaning of findable, accessible, interoperable, and reusable data, but with the emphasis already on machine actionability, into what we now call fully AI ready, abusing the same acronym. And my conviction that I really spelled out a bit more in the videos that you can play at your convenience, is that the last thing that most experimental scientists are waiting for is tools that generate millions of hypotheses, mostly, as you will see later, in the right-hand side of the sawtooth graph, where we actually perceive things as chaos because they are too complicated for the human mind to understand. And machines, especially very powerful machines, will discover lots of associations in data. You can put them even in a knowledge graph, but many of them are probably either spurious associations or not understandable for people. So there is no actionable knowledge there. So my push that I discussed with Eric before is that what we actually need is input for LLM type models that is much better than just data and text and all kinds of raw stuff that they're consuming now, frequently even violating copyright. We have to move to feeding the models with proper knowledge graphs, with provenance and curated knowledge. That's going to hopefully improve them a little bit further. But at the output side, that is more important, I think, for this event, we need to be able to constrain the output with what we call foundational ontologies or conceptual models, where we actually prevent the machine from putting out stuff that is simply impossible, like a protein can make a protein complex with a building, or the things that we consider as impossible. If you constrain the output, you will only get meaningful stuff, but still you could get very many hypotheses. My example is our own research of years ago. Just looking at genes and diseases, we found 120,000 very likely hypotheses. And we could hardly publish the paper because people said, what can we do with 120,000 hypotheses? And that was the 1 percentile score of the highest match we ever found. So what we need is to narrow down the output of machine models to a very narrow window near what I call the percolation point, but I don't have time now to explain that. It's in the video. To really restrict the output to something that people actually feel they can follow up on, like formulating a proper hypothesis, do the experiment to see whether the thing is actually true. So that was my pitch to the group, and I'm very excited to see what comes out and what ideas you have. And in the end, of course, hopefully getting some actual tools that work. So, Eric, I don't know whether you want to come forward and ask me anything that is not yet in the video, or whether things were unclear. Otherwise, I'll leave you to it and go back to my other meeting. But if you want to ask any questions, do it now.
Hi, Barend. Thanks for joining the event. First off, just wanted to say thank you for joining the event. We're very grateful to have you here as a judge and are appreciative of a lot of the insights that you provided. I think a lot of the initial information that's in the video should be a great resource for hackers as a whole. I think most of what would need to be said is actually said in the videos. Mostly we just want to express gratitude for you being here.
Okay, and I would be very interested to also, if people have questions in between when they try to build something and they really feel insecure about certain things I said, or whether they understand it well, whatever, I'm always able to answer questions either via chat or, if we need to go online again, we can always do that during the process. Okay?
Absolutely, and thank you. We may be setting up a biweekly virtual office hours or something where hackers can reach out.
Very good.
But in the meantime, Norman, I think we've got the videos queued up if we're good to go.
Okay.
All
So I
right.
wish you a lot of success, and I will follow with a great interest. Okay? Bye-bye
Thank you.
then. Bye. Hello, everyone. My name is Barend Mons, one of the judges during this event. And Eric van Winkel asked me to say a little bit about hypothesis generation and what experimental scientists like me for many years are actually waiting for. And especially also what they are not waiting for. So let's start there. The experimental scientists nowadays are not short of hypotheses. Now that we have massive amounts of data that become more and more machine readable, the machine-assisted hypothesis generation is definitely interesting. But we have done research in this area for many years. At the end of my presentation, you will see some links to our earlier papers. And if you just take genes and diseases, for example, and you run hypothesis generation software years ago, which is only getting better, we already created 120,000 hypotheses and could hardly publish that in Nature Genetics because, people said we cannot deal with this. And this was the 1 percentile score of the best matches that we found between genes and diseases that had never been connected before. So that to say, we are not waiting for enormous amounts of hypotheses that actually are too numerous to do experimental validation. We have taken 10 years to validate maybe about 6,000 gene disease associations, and each of them costs a lot of money. So it's not practical. And machines, as we will see later, see patterns everywhere. They have no idea what they mean. So finally, we need hybrid intelligence for people to come back and check the hypotheses and then prove them. And if that is so massive that we have to plow through enormous amounts of potentially meaningless hallucinations and spurious correlations that the machine sees, that's absolutely not what we are waiting for. So what we would actually need is tooling to zoom in on where the actionable knowledge is, meaning new insights that were not seen by people before, but the machine is much better in pattern recognition, so you see new patterns, and there must be a high likelihood that those hypotheses make sense in two senses, that they can be validated experimentally, and that they make sense in the conceptual model of reality that we have, for example, in health or agriculture. And a very important note to start with is that we need knowledge graphs, and preferably curated and provenance-rich knowledge graphs to feed models, because just feeding them with all kinds of text, also knowing that even the peer-reviewed scientific literature contains a lot of outdated or actually false information and claims, even the peer-reviewed literature, mind you, if you feed those to LLMs, they won't be able to figure out what is true and what is not true and generate a lot of meaningless hallucinations. So what we see now is that data becomes so big and so complex that we cannot share them effectively, also because many of the data are not even shareable in the sense that they're highly sensitive. So we continue now with data visiting, which you see here is an animation of what we call trains, algorithms that go to data stations. They ask permission to do a certain study. They only take the results with them. We call that data visiting. And of course, machines, being rather stupid, need to understand the data in the station very well. So the data should be findable, accessible, interoperable, and reusable, the original meaning of FAIR. But obviously, we now rebaptized FAIR also as fully AI ready, because if the data are not understandable and actionable by machines, without the help of people in most cases, you will get total mess. So why is it so important to have machines independently using the new Internet of Fair Data and Services? Well, first of all, you all are very familiar with the many V's. I just mentioned volume, variety, and velocity. We have many other V's now, but that is what we essentially deal with when we deal with big amounts of data. And if we look in my field, the health care and health research, we estimated that meanwhile, in the literature and everything that we have produced, we have around 10 to the 14th assertions. Well, good luck to run a Sparkle query over such a graph. I don't have to tell you, I suppose. Meanwhile, what we see is that the data are hidden behind articles. So it's horrible. The machine needs to go to an article, and then if they're lucky to find a link to data, they're usually PDF files or totally useless data formats. So a lot of the data, although there is a link in the original paper, they appear to be at the laptop of a PhD student that meanwhile went back to China or whatever, and are simply not findable. We estimated, or probably there's even some proof now, that about only 27% of papers are really leading to the data that support them. That's another problem. Now, meanwhile, in my field, genetics, we double our data every 6 months. That's exponential. So there is only one way to deal with them, and that is with machines. But that brings a lot of challenges with it. First of all, also privacy. And privacy, don't only think about the data on people. So you have, for example, if Naturalis, our natural history museum, would publish the data on the last rare plant in the Netherlands, it will be picked the same afternoon. If in India, the photographs of tigers on snapshots are published, they will be dead the same afternoon. When the mussel banks in the Netherlands were published, they were fished empty the same afternoon. And of course, in some cases, we also don't want to find out what is the identity of a patient. But that's not the only thing. Then you have, obviously, complexity. And what we have to realize is that machines, also LLMs, although they give very, very impressive output, don't get me wrong, in essence, the machine has no clue what it's putting out. It's just doing very advanced statistics and modeling, machine learning, and then there is an output of words. And essentially, in that sense, everything is a hallucination. But we project value on it when the hallucination makes sense to us. So either is a known thing or a credible hypothesis. That's the same what we do with animals. We project all kinds of human-like features on animals. Now we start doing that on robots, but essentially, I would say for now, and it's a bit controversial, but that's fine for a hackathon, that LLMs and likewise other models are just spitting out hallucinations. Some of them are useful, many of them are totally useless or even misleading. So how do we deal with this now? First of all, we need to feed the model wherever possible with fair knowledge graphs that are high in provenance. So even machines could find out that certain triples are no longer valid or are highly contested, like hydroxychloroquine treats COVID. It is published many times in papers. So if you use input papers, you'll find that triple many, many times. But we know that is now highly unlikely, if not proven untrue. How do we tell machines, don't take that triple seriously? And on the output side, the right-hand side of this slide, we need to constrain, and that's the most important part for this hackathon type activity. We should be able to constrain the output with our conceptual model. I'll give you one example. We should not allow the machine to suggest that a protein can make a protein complex with a building, which is, principally impossible. But the machine would easily do it if it is not told where the conceptual boundaries are of what it can output. Given what we already saw in my brief introduction of 10 minutes before, this is probably the most crucial slide to understand. Eric van Winkel already made some comments on this, but let me walk you through the system in the context of what we are discussing here. So this is a model that my group made years ago, where on the x-axis here, we actually plot the complexity of patterns, starting with 1, 0, 1, 0, 1, 0, to very complex patterns. And at the end, we have maximum, complexity of probably the whole world. Then on the y-axis here, we have assumed a certain level of understanding, let's say intelligence, CPUs, computer understanding, the ability to discover all the patterns that are in the data set or in the data that are on the x-axis. Now, obviously, when you go more and more complex and you keep adding understanding, probably the line goes straight because you keep understanding all the patterns that are in your source data, whatever they are. Obviously, over time, this line, I will explain the percolation point later, moves to the right. We understand more and more things, we make more and more assumptions and discoveries. So the patterns of real life data that represent our knowledge become more and more complex. But to a certain extent in history, there were still universal geniuses that knew everything humankind knew in science at that time. Well, we all know that time is far behind us. And coming back to the idea that we have only in the life sciences about 10 to the 14th assertions to deal with, there's obviously no way that humans can further increase their knowledge at some point, individual humans. Now, of course, we can put many people together in big networks, but we cannot connect their brains effectively. Differently a little bit with computers, where we can make very big supercomputers and so on. But at some point, you see this line, the addition of understanding stops. There is a limit. At that very point, a very interesting phenomenon is taking place, which creates a perfect version in the model of the famous sawtooth graph that you see in a lot of complex systems. And what is happening is the following. You get a further increase of the complexity in the system. And suddenly your understanding goes asymptotically to zero. So a little bit more complexity and you lose control of what is going on there as a human for sure. If you could add more compute power or more understanding, probably the line would go straight up. Very interesting philosophical thing is that in the end there would be no chaos, because very much we experience what we see on the right-hand side of, let's say, the red line. We are somewhere between the percolation point and the red line now, as chaos. You don't see the patterns, or at least they don't make any sense to you. Now, one of the projects that I can take as an example that I'm involved in as a scientist is to understand the human immunome, which is a combination of what we call, the genome, our DNA, the transcriptome, our messenger RNA, proteome, the proteins, all the small molecules in our body, the microbiome, the bacteria in our guts and everything. Then how the genes are regulated by epigenetics, and finally also how the environment, the exposome,
is influencing this incredibly complex system. To get insight in that, we absolutely need computers, but if we let computers go rogue, they will give us an immense number of assertions that we cannot deal with. Where we actually discover actionable knowledge is right here around this percolation point. And if you then go back to the idea of the slider or the parrot that we discussed in the previous video, you could say if you would have a slider to say, I put a very strong constraint on the output of any machine learning model that I had, then you would only find, solid knowledge in ontologies that you already know. That's probably what you want when you are dealing with decision making in health, for example. But what we are talking about here is really hypothesis generation to gain new insights that people have not discovered so far. So then I would put the slider somewhere around the fluid point or the percolation point here and move it slightly to the right, where you will get new correlations and potential associations, hypotheses that would still make sense to people. If you would put the slider fully to the right, allowing maximum hallucination and serendipity, you would get all kinds of crazy correlations without any restriction, and we would consider that as chaos. That is the least we are waiting for. So if you want to make a hypothesis discovery tool, basically what you have to do is to make sure that people, we are talking hybrid intelligence, can put a slider at a position where they still consider the output of the LLM, although new, LLM or any other model, to be meaningful in the sense that it is an hypothesis that they could A, test, and B, that makes principal sense, like a new drug for a disease or something like that. But total crazy stuff that is probably even true. One of the examples that I always give is that if you put the slider far enough to the right, the machine will tell you that 25,000 genes and 4 million regulatory sequences in our genome make us human. Very true. But we cannot do anything with it in terms of actionable knowledge, which gene is causing which disease or anything like that. So there is this window around the percolation point where knowledge discovery takes place. This is really the theory of knowledge discovery in a way. So that is a very nice challenge for this hackathon, I think, to try and find a way to constrain the output of any model with a conceptual model-based filter, slider, up to you how to implement it. That would help scientists to find meaningful, testable, and logical hypotheses. Hi everyone, this is the third video I would contribute to the hackathon. And this is about an approach that we have taken and that could be the basis for much of what you can be doing. So 10 years ago, we came up with this acronym FAIR for findable, accessible, interoperable, and reusable data, with an emphasis on machine actionability. Obviously, as you see in the center here, AI is almost part of that acronym by default. And now we have also taken on the meaning of fully AI-ready data. And this is very relevant for what we discussed in the first two videos. Why? Because if machines relatively independently from people have to visit data and make sense of it, the machine should at least know what the data mean. So there should be no ambiguity. There should be a very strong provenance. So FAIR is really the basis for responsible, explainable, and future AI that really, really makes sense. So let's look a little bit more about this whole approach. If you think about knowledge discovery and what data should support, We deal with what we call concepts. That could be a disease or a person or anything you can talk about, and that is a unit of thought. Obviously, in the Ogden triangle, we need a token in human language, but also in computer language, to refer to that concept. Like, let's take cancer here. You can call it malignant neoplasms, Krebskrankheit in German. There is also a CO265, which is the code in the Unified Medical Language System, etc., etc. So there are a very long list usually of literals or URIs that point to a concept. And what we need in FAIR is that the identifier, whether it's a literal or a machine readable identifier, should always resolve to the intended defined meaning. So if you say bank and you don't know whether you mean the financial institution or the riverbank or a piece of furniture, then the machine has no clue unless it can derive this from very complicated context analysis. So we need to have what we call a GUPRI, a globally unique, persistent and resolvable identifier for every concept that is in your graph. Not only the subjects and the objects, also the predicates should be well defined, and the URI to point to them should always be only resolving to the intended defined meaning. And this is irrelevant whether the object really exists as in touchable in reality, because a concept like love or even cancer, a disease, is not touchable. It's a mental construct. But still it needs a unique identifier, a good definition, to ever make machines able to deal effectively with data. Because machines are very stupid, we need to tell them very exactly what's going on, including provenance. Otherwise, they will come up with massive amounts of hallucinations. Now, one of the problems that ontologies cannot solve is what we call near sameness. We as humans are very good in disambiguating on the fly when we communicate. Like we say, I love the piano. Most people will immediately know that this is piano music, not necessarily loving the instrument, or a floor in Italy, or soft music. So, and in many cases, even if we have a near same concept in our mind when we communicate as humans, that's okay. However, if I'm a mover of furniture, I would probably ask a few more questions. Is this a grand piano or an electrical piano? That makes a big difference, but not in different contexts. Now, machines are horrible in dealing with near sameness. And one of the mistakes I see a lot of computer scientists make, in my biological mind, they call it the stupid biology version, is they say, okay, now if we have to do hybrid intelligence, where we bridge the gap between humans and machines, what we are doing is a lot of work to fill these gaps that cannot be filled because a machine is not a human. So don't try to make everything smooth, completely understandable, but go to hybrid intelligence where the machine is good enough to give people very meaningful content that they then shape to final human knowledge. Machines have no knowledge. Machines put out patterns. We have a way to do that and say a machine should be able to, independently from humans, or at least as independently as possible, to deal with near sameness of two concepts, like two proteins, one is in the mouse, one is in the human. They're almost the same, but not quite. How do we tell computers that without having an incredibly complex ontological approach to that, which is going to fail, in my view? So that all comes to a very basic concept that we started in 2009. It's a nano-publication, essentially a single triple or a small graph that is composed of single triples, subject, predicate, object, green, yellow, red. It should, as a whole, have this globally unique identifier, GUPRI, because the whole assertion is a machine actionable units of information and therefore a concept in the Ogden triangle, and it should have rich provenance. Who said it, when, where, how was it created, like a scientific assertion. Now, as we said, we estimate probably that we have about 10 to the 14th of those nano-publications if we would be able to gather them from anywhere in the world. And some companies have done a good job to gather the most important ones. Then, of course, if you have a triple like hydroxychloroquine treats COVID, that was published in the Lancet, very important journal, in 2021. Meanwhile, we know it's not true, but hey, it's still there in the literature. Maybe there are a lot of people that have contested that triple. How do we feed machines with that information, for example? Now, one of the things that we did, we have so-called cardinal assertions. There's many times that you find the same triple. Hydroxychloroquine treats COVID. Different people said it, so it gets a new identifier. So the machine knows there are many iterations of this thing. The provenance is different all the time, different author, different time date stamp. But if we bring them all together and we say we store the basic triple only once, and we have a large number of provenance files, maybe somewhere in a different database, so it doesn't disturb the graph, you already go down to 10 to the 11th, more or less, triples that are what we call a cardinal assertion. So the cardinal assertion, hydroxychloroquine treats COVID, is in the literature and also in a proper knowledge graph, but it should be annotated in some way as not being valid anymore, or at least the machine should be able to say, hey, there's 800 scientists with their Open Research and Contributor ID, the ORCID, that have contested this triple. Doing that would be to create another triple saying ORCID, subject, contests, predicate, this triple. The triple has its own GUPRI, is a concept, so now it can be annotated. Now, the next step is, if you take all the cardinal assertions that start with the same subject, like hydroxychloroquine or malaria or COVID or anything, you can create what we call a knowledge or a fair digital twin of the concept of a disease or a drug or a gene or whatever. And again, if it's composed of machine actionable units of informations like cardinal assertions, fair digital objects, the whole new container is again a graph with one concept in the center, but it's again a fair digital object with its own GUPRI and its own provenance. That's very important to understand. Now, if you have such a knowledge and it has its own GUPRI, it can be followed. Now, a nano-publication is essentially immutable because it was set at a certain point in time, but a knowledge, think back of the sawtooth graph in the previous video, a knowledge would change basically every day if you get more insight into a disease or a drug or a person. So knowledge should be traceable over time in a sort of blockchain-like fashion. I'm not pretending it solves a blockchain problem in all aspects, but it is important to understand that knowledge will move through conceptual space on a daily basis if there is one or more triples added to it or contested or whatever. Now, in this way, you can, as it were, zip the explicitome of everything that is out there in the literature. And let me tell you, I don't believe at all that LLMs will sort of make this obsolete or solve this issue at all. So we have 10 to the 14th associations or nano-publications. We have 10 to the 11th cardinal assertions with n different provenances. And we have only about 10 to the 6th, a million or so knowledge right now in the life sciences, if you restrict it to important concepts like genes, diseases, drugs, metabolites, etc. So that's manageable. Actually, this whole graph would fit on a laptop. Now, what you can do is say, hey, here, now we come to the knowledge discovery and hypothesis generation. Here we generate, for example, that the knowledge of malaria and Tegafur, a certain drug, are very close in conceptual space. And they actually move closer over time. At some point, they pass a threshold, and you say, hey, Tegafur, a drug that was originally made for cancer, could work for malaria. First thing is, does this make sense? We don't believe it just because the computer suggests it based on closeness. And here you see that Tegafur inhibits a certain enzyme that is also a target for an existing drug in malaria, pyrimethamine, that kills malaria parasites via this enzyme. But the malaria parasite is resistant against this drug. That's what we know as humans. We can also have that in the graph. Now a human can decide this is a potentially credible hypothesis, so let's test it. We actually tested it. Tegafur is too large to get into the red blood cell, wasn't in the graph, so humans had to decide, very nice idea, but practically impossible, because we would have to make the molecule smaller, and then it has to go through the whole FDA approval again, and that's too expensive for a malaria drug. Now, obviously, we would be able to also take some triples out of a knowledge because they're irrelevant. Take the example of two proteins. One is mouse, one is human. They are identical up to the amino acid, but one is produced in the mouse and the gene is on a different chromosome. Then two triples in the two knowledges are different. So the machine knows a number of things. Because the goopri in the center is different, the machine knows, quote, knows, those proteins are not entirely the same. Secondly, it knows the small difference, different species, different chromosome. But if I say, I don't care whether the protein is made by a mouse or a human, I just want to look at its structure for now, I can create what we call a qua by filtering out these concepts, that the objects in the knowledge that are of a semantic type that I don't care about, like species or chromosomal location. That is how we can tell machines with conceptual models and quas to treat a certain concept in a certain way during a discovery process. And these things move over time. So we can now start, and we did this years ago, show that a gene is consistently moving closer to a disease, for example, in the conceptual space of 1,000,000 by 1,000,000 concepts, even way before humans have written the first article saying, hey, this gene may be involved in this disease. So knowledge change over time. What you can also do now is, if you have very complex knowledge, these are real knowledge, but already filtered like crazy. So these are quas already. We only look at proteins and metabolites and so on, between three major diseases: metabolic syndrome, diabetes type 2, and early onset Alzheimer. And there seemed to be a correlation. By just throwing these three knowledge together, in, of course, a machine actionable environment like Euritos, that's a Dutch company that runs this kind of stuff, you see that lots of concepts are in common between the two major knowledge at the top. Less so the early onset Alzheimer at the bottom, but 5 concepts in the middle are really associated with all 3 of them. And that means that those are probably the enzymes and the metabolites you want to focus on. And indeed, we find already literature where this makes sense. Again, computers suggest a hypothesis. We want to see the graph. We want to see the provenance. We want to see the rationalization. And it could make immediate sense to experts in the field. We can also combine databases that are in nano-publication format that have never been working together before. Here you see, for example, a typical nano-publication from the Leiden Open Variation database in green, where doctors say, hey, I find this strange mutation here in the genome. So in the center, you see numbers, which are just the positioning in the genome, in a chromosome. Of course, you have to use the same reference model, reference genome and everything. Here you talk about human chromosome 19, chromosome 2 of a particular human genome. And you say, hey, in a totally different study, we found out where so-called transcription start sites are in the genome. This is where genes start their subscription, and dependent on that, you make the right or the wrong protein. And we showed very easily that more than 1,000 known disease-causing variants do not map to the genes at all, but they map to these transcription start sites, which probably results in making the wrong protein in the wrong tissue, and that's causing the disease, not because the gene itself was affected. So you see that all this kind of complex hypothesis generation cannot be done without the computer, cannot be done effectively without nano-publications and graphs. But always you need the human intervention. This is one more example from adalimumab, the monoclonal, that caused hypoglycemia in some patients. We know it inhibits TNF and also other stuff. And we found very clearly that maybe the reason for that side effect could be insulin resistance in some patients. Just another example. Now, a very complex model that we created for COVID in those days, you can see that you need all these cardinal assertions. Each of the links here is a cardinal assertion, and if you click on it in the live version, you can see why and how many people have said it and so on. So it's very complicated stuff. We can throw all the drugs that were in panic used for COVID to see whether they worked and predict whether they made any sense in this immunological model. And for example, here in the top, you see hydroxychloroquine with only one line to one of the important proteins. So we predicted early on that would not be a good approach. While other drugs that are still very much used and studied, like dexamethasone, was highly connected in this data model. Again, this is what we call a ridiculogram for people. They cannot deal with this. Machines can, but we have to be able to extract the hypotheses that make sense and are close to what we call the percolation point in the previous video. And if you then come to, okay, why would tocilizumab, for example, a Roche monoclonal, work against COVID in certain cases? Okay, you know what it interacts with, and that makes perfect sense that it could work in the case of COVID. And here you see that it is supported by 7 publications, and that is part of this TNO project together with Euritos. And of course, the whole noded is a ridiculogram. But if you extract the potential hypothesis that you can test, and we have actually tested it later, that makes sense. And that is what we are waiting for. And of course, the parroting or the hallucinations or the patterns that machines discover are useful, but they always have to be checked by good input, a conceptual model to constrain them for hallucinations. And humans will then have to look at the results, could be in Zoom like this one, and you actually then come to a conclusion with a doctor, with a bioinformatician and so on. Does this make sense? And that is my last point here. Coming back to this famous graph, what we actually would need is tooling that allows us to go from solid knowledge just for decision making, potentially all the way to chaos and complete nonsense, but somewhere in the middle, there is this holy grail where we find new knowledge that is actually original and new, but also actionable in the sense that the hypothesis can be tested within the boundaries of financial constraints and experimental possibilities, and it makes sense to experts in the field. And of course, sometimes the expert may say crazy in the beginning. The further you go to the right, the more you will find that. But a lot of scientific discoveries in the past were originally considered very unlikely, and they were still true. So this is what experimental scientists are in need of. And if you can give us any leads in this direction, you would be our hero. Thank you very much.